Okay, good morning. Welcome to the webinar this week. This week we're going to be talking about remote sensing methods, measuring canopy and energy with a UV-VIS NIR spectroradiometer. So my name is Mackenzie Woodman. I'm the application specialist here at Spectral Evolution. And yeah, today we'll be talking about canopy measurements. Thank you all for attending. So first off, I'll start by introducing Spectral Evolution. We're the leading manufacturer of high-resolution UV-VIS NIR spectrometers and spectroradiometers for remote sensing, mining, and laboratory applications. We were established in 2004, so we just celebrated our 21-year anniversary, and we're located in Haverhill, Massachusetts, so just outside the Boston area. So what is a spectroradiometer? And what is spectroscopy? So spectroscopy is the study of the electromagnetic spectrum as emitted or reflected by various materials. The electromagnetic spectrum is a representation of electromagnetic radiation, and they're defined by their wavelength. A reflectance measurement directly relates to the material's chemical structure and bonds. You can think of your eyes kind of like a rudimentary spectrometer with a much smaller spectral range, so just the visible light. So we take light in and we measure where it is on the electromagnetic spectrum, and from that we can determine colors, whereas our spectrometers are hyperspectral, which means they measure on each nanometer, and they're able to measure from 350 nanometers to 2500 nanometers for most of our models. So when characterizing canopy, I just want to define some of the terms that will be used for this kind of application. So canopy is the forest top layer formed by mature tree crowns, controlling light and weather exposure and supporting diverse wildlife in the top of a forest system, so the canopy structures. The ground foliage consists of all the low-lying plants that thrive in shade and help protect and stabilize forest floors. And then underneath that, you have the soil or nutrient-rich foundation that supports all of that plant life, regulates water, drives decomposition and nutrient cycling. And then within all of this, we have a diverse array of plant species, from the big trees to the mosses and any shrubs in between. So these create different structures and different systems with the light. Forests are kind of a competition for light. So there's in nature, there's always a little bit of competition for resources. And so that's what's going on here. And then under that, we have that middle ground, which is the understory that lies beneath the canopy, which includes like younger trees, shrubs, things like that, that will also be competing for light and energy. So with satellite and drone applications or remote sensing applications, reflectance data is collected above the canopy. So by using a field spectrometer, we can actually go underneath the canopy and start to characterize how this forest system is working. The primary portion of remote sensing data that's taken above canopy is going to be just reflectance measurements off the top of the canopy. And so there are other wavelengths that can penetrate canopy, but as far as the full range, this would be the option to, or using a field spectrometer or spectroradiometer would be the option to get sub-canopy measurements and characterizing it, the energy balances there. So there's limitations to that satellite and drone application. There's also challenges to going out in the field that are very important that we've incorporated into the design of our instrument. So Having an uncontrolled environment is very important. Environmental conditions are rapidly changing. If a cloud goes by the sun, if your conditions start to change, like your light conditions, because oftentimes it's a ratio between the light that's coming down and the light that's reflected off of an object. So characterizing that is important, and making sure you're taking references quickly is important. So our instruments are designed to be reliable and stable, and as stable as possible, so that you can quickly react to different changing conditions. They're also field ruggedized, so that you don't have to worry about the instrument breaking in the field typically. And then for a field campaign, you're going to be worried about how accessible is the location, what is your portability of your instrument, how much power are you going to need, how many batteries do you need. So all of that is incorporated in our design. The new NaturaSpec Plus has an internal battery source, so that you can quickly hot swap batteries, never actually disconnecting from your computer controller or tablet for field use. And then for lab quality data, we have some of the best spectral resolution, signal to noise ratio, and very important optimization steps. So as soon as you hit the target button, it will go through a range of optimization steps that you won't need to regulate. They'll just be done on every target measurement. So when you hit the target button, it will take a dark measurement. Then it will take a quick scan and determine the optimum amount of integration time to each detector, so that you get the maximum amount of energy to each detector without saturating it. And then it will take your measurement on whatever averaging you have. So it could be 30 averaging, which means it'll take 30 scans and average them together and give you one scan. And then after it, it will take another dark measurement and average the two dark measurements before and after and subtract that off to get rid of any instrument noise. It's called a dark correction. So by doing this, we're able to create some of the highest quality data in spectrometers and field spectroradiometers. So what are the technology trends in remote sensing? We're always striving for increased spatial resolution, spectral resolution, and temporal resolution. And then, like I was saying earlier, there's little ability to characterize sub-canopy. And you'll see that when we do the energy balance study in a later slide or example. So we're constantly striving towards hitting or characterizing smaller targets, increasing the spectral resolution, what we can get away with. So moving towards hyperspectral from multispectral sensors, and how frequently can we revisit the site. So with a field spectroradiometer, you can visit as much as you can get a person there. With the satellites, you may be waiting a few days. If the weather's not good, then you'll be waiting weeks. So that's something to think about when you're designing a study. Or drones, you can fly them whenever you need to, but you may need the conditions to launch the drone if it's a fixed wing or something like that. So with our instruments, we're able to easily change our field of view and govern what size we're scanning. So you can change how big your field of view is at different distances by simply changing the lenses that we have. So we have a wide range of different angle lenses. They can all be radiometrically calibrated to get energy units. And for this example study, we used the inline diffuser, which comes with a, it either comes with a protective lens or not, depending on your preference. For this, we used a windowed diffuser, so that it has a little extra protection for in the field, and for different kinds of applications, it just has extra protection. And with this inline diffuser, it simply connects to the end of your fiber with an SMA connection, and it will give you a 180-degree field of view. So I use this lens along with the sensor probe to get my measuring angle. and I was able to collect horizontal irradiance at below canopy and above canopy using this method. So, what is the forest energy balance? That is the incoming solar radiation intercepted by the canopy, where a portion is reflected, absorbed, or transmitted, and then it reaches down to the lower vegetation layers and the ground. So the canopy reduces the amount of direct sunlight reaching the ground, alters the light quality and quantity available to understory plants, and affects the energy balance. Like I said, this is that competition that happens in nature for resources. So depending on the ecology, canopy can create microclimates underneath. So if you've ever been under canopy, it's typically a lot cooler under the canopy than it is out in something like a parking lot or even an open grass field. You'll see a big difference in the humidity, temperature, and energy underneath. And this graph on the right shows basically that as you get an older forest, you get the relative light intensity much higher up in the forest. So you get the taller trees getting most of the energy and not reaching the underneath. And so that can affect things like biodiversity and things like that. But that's the system, and we're characterizing it. So things that can change the canopy are things like forest fires. They often destroy or thin the forest canopy, allowing for significantly more light to penetrate to that lower group of vegetation. And by doing that, it creates a warmer environment with more sunlight available, which helps it to regrow afterward. So it's part of that system, and that's something that could be characterized. So it changes the albedo of the ground as well, which further warms it, so you don't get that microclimate underneath the canopy anymore. And it changes the composition of the soil temperature and moisture and evapotranspiration underneath. So it's a very important dynamic system that happens in forests, and something that could be further explored with a spectroradiometer as well. So an important dynamic with that is albedo. So those ash floors will have a higher will have a lower albedo, I should say, more absorbent to energy. And so that will increase the temperature there. So albedo is the measure of how much sunlight or solar radiation that is reflected by a surface. It ranges from 0 to 1 because it's a ratio of the upwelling energy divided by the downwelling energy. So yeah, that's important with creating those microclimates, understanding that. So another thing that can drastically change the canopy is invasive species. They can outcompete native canopy trees, alter light availability, and reduce biodiversity. So the changes in the canopy from invasive plants or bugs can lower or increase surface albedo, affect the local energy, and it may change what plants actually can live in that ecosystem anymore. So it'll change evapotranspiration rates, disrupt water or heat exchange, and then you lose the native canopy sometimes, depending on the invasive species. And so you could design a energy study showing just how much some of these plants, like things like kudzu, which is a great example of an invasive species that drastically affects canopy. So basically how it works is it will completely blanket an ecosystem and choke out all of the ecosystem underneath it, creating a monoculture. So understanding that relationship of different species of plants and how light affects it and how it's spreading is also very important, and something that would be a great use of a spectroradiometer. So here I have collected some data just to show how quickly you can get quality energy data underneath a canopy. So these higher energy lines are from an open field nearby where it was forested. The lower energy lines are sub-canopy. So those are measured below the canopy. As you can see, there is some shifting between them, but before calculating energy, I took the average of all of the scans that we took under the canopy, and that was just due to either clouds or some shift in the energy getting to where I was sampling sub-canopy. So there's some differences between where you are in the forest and that. So we took the average to calculate all of our energy, and all of it was done at zenith with a cosine diffuser. And we used the integrated energy tool in Darwin, which is in all of our Darwin software to calculate these energies. So each spectroradiometer, the NaturaSpec series, remote sensing series of instruments, they all get a radiometric calibration. It allows us to convert detector counts to energy units. And then with this integrated energy tool, I can select scans and actually measure or calculate the energy across different parts of the spectrum. So, and I can select what units I want to be working in as well. So the above canopy irradiance was measured at 689.87 watts per meter squared. Below the canopy was 64.19. Now this is the average of what we collected in the field. It was, and then so from that I can see that it just left 9.3% of the ground foliage light. So, but that's not where the energy assessment ends. We can actually decompose this into different parts of the spectrum and start to see where the vegetation is absorbing the most, where is it absorbing the least, and what's the dynamic here. So in the UV VIS, we get about 8%. In SWIR1, we get 15%, which is slightly higher. I'll double. But still a lot less energy. And then in SWIR2, we get 7%. But as you can see, there's also significant energy differences. As you can see, UV VIS is the primary portion of the energy in the system here from the sunlight. So it's an interesting way to look at energy and light balances, but what studies can be done with this kind of data? So energy in different forest ecosystems and evapotranspiration studies can be conducted, quantify effective seasonality on ground cover. So you could collect data every day or once a week for some seasonality differences and how the energy dynamics work. Is there more light available in the springtime or fall for new growth? Just something that's interesting. I'm sure fall obviously would have a lot more light because there's less leaves, but in an evergreen forest, obviously that wouldn't be the same relationship. So characterizing that is important. Energy balance through forest maturity would be great. So forest maturity is very important for biodiversity, or just a biodiversity study is also rewarding. And then recent studies seek to incorporate leaf level spectra into modeling and machine learning. So really getting into the nitty gritty of these light dynamics. And so when doing that, you can use our leaf clip to get leaf level spectra and lab quality leaf level spectra. So the pine needle adapter also incorporates onto this leaf clip probe, which will allow you to target even smaller and thin pine needles. So with this leaf clip, what we've done is we have a tungsten halogen light source internal to the leaf clip, and that fiber optic you can see at the top actually takes the light source away from the sample, ensuring that we don't dry out the leaf or burn a hole in the leaf. It takes out that thermal component. And it sends the light through that fiber optic onto the leaf. It also has that white plate there for ease of use. And using this, you can get lab quality data and characterize all the different species in a system, and then see how that relates to how much energy is really penetrating underneath. So it's a useful tool for these kinds of measurements. And that was my cursory overview of Canopy studies using spectroradiometers. In conclusion, Spectral Evolution's emphasis on field capabilities and lab quality allow researchers to experiment with a diverse set of applications at a sub-canopy level. There's a wide range of studies that can be conducted on the energy balance and ecology of forests using the NaturaSpec series. I'd encourage you to do so, and Spectral Evolution sets the standard for high-resolution canopy measurements. Thank you very much, and I'm happy to answer any questions on this study. Thank you.